Thank you very much for coming to my talk. This is a story that I would like to present that represent our effort in profiling patient data, which has led to the discovery of a new activity of what appeared to be a very familiar tumor suppressor, NF1. Most of us have nothing to disclose except the individual listed on this slide. Now, NF1 loss at the germline level is responsible for neurofibromatosis type 1, the world's most common genetic disorder. The best understood function of NF1 is to function as a GAP GTPase activating protein, which function to hydrolyze the GTP bound to RAS. So when NF1 is lost, or when GAP is lost in general, RAS is stuck in the GTP inactive state, leading to constitutive signaling downstream. However, recently, when we examined breast cancer cohort treated by tamoxifen monotherapy and followed for over 15 years, we found that while NF1 mutation readily correlate with poor outcome in this cohort, the NF1 mutation identified here are all nonsense and friendship mutation. We did not detect any GAP mutation that inactivate its GAP activity. Thus, if NF1 is a GAP, the GAP activity does not appear to be important for the apparent induction of tamoxifen resistance. This has led us or forced us to think outside the box and leading to the discovery that NF1 turns out to be a direct and canonical ER transcriptional co-repressor, and it can act independent of RAS activity. So the model that we propose is on the left, that's published several years ago in Cancer Cell. But I think the experiment on the right best illustrates our effort to separate RAS activity from ER repression. So we make two mutations and measure ER transcriptional activity. The red genes are those that are usually induced by estrogen. And when we knock out ER binding site, shown here in red, the cells appear to express these E2 responsive gene even in the absence of added estrogen. Whereas when you inactivate GAP domain by the mutation mark in blue, they behave like wild type. So the model on the left suggests that when NF1 is lost, ER transcriptional activity goes up. This leads to resistance to tamoxifen as well as to aromatase inhibitions. Our data suggests that for this type of tumor, the best way to treat them is to use a SERD, selective ER degrader, such as fulvestrant, which lead to the degradation of ER. And then you have to block the RAS pathway by a MEK inhibitor, which prohibit the feedback activation of survival pathway of ER positive cells when their major signaling pathway is inhibited. The preclinical model for this hypothesis is shown here. We have a collection of many ER-positive PDX models, and we found at least two models shown here in red are actually NF1 deficient. We developed an IHC assay shown here on the left, and then we also developed a mass spec analysis, so we can actually quantify the level to define a threshold below which we have these two models that when you treated them with fulvestrant plus binimetinib, which is a MEK inhibitor, as shown on the right, you readily detect tumor regression, whereas the tumor that has detectable NF1 only show growth inhibition. Based on these preclinical studies, we are currently conducting a clinical trial as part of the combo match trials. So in an effort to find out what else may be vulnerable in NF1 low ER positive tumors, we examined the phosphoproteomic database in the CP-TAG cohort, which contained about 150 breast tumors. We compute a kinase activity score based on the phosphorylation pattern and ask the question, how does that score correlate with NF1 level? Shown here in the left side of the volcano plot, we readily detected elevation of CDK4 and CDK6 activities in NF1 low ER positive tumors. And this piqued our interest because CDK4 and 6 is currently the standard of care together with the endocrine therapy agent for ER positive breast cancer. In the cell line model, as shown here by the Western blot, you can clearly see in the cells the PRB levels also go up. As you may know, phosphorylation of RB is the predominant substrate for CDK4 and 6. We also measure phosphorylation of the activation loop in CDK4 at the position of 172, and we also detect phosphorylation at that site. So indeed, NF1 loss appear to activate CDK4 and 6, leading to downstream phosphorylation of RB. What's the mechanism of this regulation? We know ER is recruited to the cyclin D1 promoter, and NF1 as a co-repressor. We can show that when NF1 is lost, this site where ER is occupied more by ER. So the recruitment of ER to directly induce cyclin D expression is greatly enhanced due to NF1's loss as a transcriptional co-repressor. Also, despite the fact that it's known CDK4 has to be phosphorylated at the position T172 to be active, what kinase pathway may be responsible for this is not clear. Interestingly, we have evidence that this phosphorylation appeared to be RAF dependent when we treated the cells with a pan-reactive RAF inhibitor. So what is the clinical significance of these studies? We know that shown here is a collection of overall response for a number of clinical trials. Even in the first-line metastatic breast cancer setting, on average, about 30 to 40% of the patient will respond. The great majority of the patients do not. We do not have a good biomarker to predict which set of patients will respond, which set of the patient will not. Currently, if you are ER positive, you are eligible for CDK4 and 6 treatment. And if you are RB negative, probably it's not a good idea that you will be treated by this therapy. So how does the NF1 status can impact our decision whether or not to treat the patient with CDK4 and 6 inhibitor? So we turn to our PDX model again. As you remember earlier, we have two models, Wing16 and Wing24. They are basically NF1 null as determined by IHC. And here again, these two models readily regress when you treated these models by fulvestrant and palbociclib. The great majority of the ER positive model that we have seen, the best you can get is growth inhibition as shown in Wing40, which is a model that has median level of NF1. RB status is one that potentially can mediate response to CDK4 and 6, and all of our model has normal level of RB, including Wing40, and Wing40 respond perfectly to the treatment of fulvestrant as well as palbociclib. Shown here on the left is that we can also detect apoptosis in the cell line model. Here we use abemaciclib. So no matter which CDK4 and 6 inhibitor you use, the response is similar. All right, so can we detect similar to determine how NF1 status can impact the efficacy of CDK4 and 6 inhibitor? We examined the NeoPanana trial in which the patients were first treated by a aromatase inhibitor, enaclazole, followed by the addition of palbociclib. The tumors were divided into NF1 high and NF1 low based on mRNA levels. As expected, the NF1 high or normal tumors in the presence of aromatase inhibitor, you can readily see a reduction of proliferation as measured by a multi-gene proliferation score, whereas the NF1 low tumor did not seem to respond substantially to this treatment. However, great proliferation decrease was observed when palbociclib was added. The impact on apoptosis was measured on the right, in which apoptosis score were plotted against NF1 level as a continuous variable on the x-axis. And here you could see that in NF1 low tumor, the treatment of enaclazole actually have a low apoptosis score. However, when palbociclib was added, the apoptosis greatly increased. So these data agree with our preclinical studies that a subset of NF1 low ER positive tumors are vulnerable to CDK4 and 6 inhibition. So at the end, I would like to entertain a model. NF1 loss leads to the activation of both RAS and ER is known to activate this pathway by controlling cyclin D1 expression. The RAS and RAF pathway is also known to regulate expression of cyclin D1 through a different transcriptional pathway. Furthermore, RAS and RAF is necessary for cyclin D1 binding to CDK4. And now we have evidence that perhaps the phosphorylation of CDK4 itself is also dependent on this RAS and RAF pathway, leading to the full activation of CDK4. It is possible that CDK4 and 6 activation is the key survival pathway for ER-positive breast cancer cell under the control of RAS. This makes NF1 deficient ER-positive breast cancer cell particularly vulnerable to CDK4 and 6 inhibition. To thank many of our collaborators on this project, those that played direct and a leading role are shown here in bold typeface. In particular, I would like to point out Bora Lin, who is my clinical collaborator leading the clinical trial. Thank you for your attention. I'm ready for your question.